Hi all, welcome back. This is chapter 16. We're going to talk about how bacteria regulate their gene expression. Bacteria generally regulate their gene expression all at the transcription level. So I want you to review transcription and translation before you start, but particularly transcription, because this is where this entire lecture is going to happen, where we're going to decide if we're actually going to make a transcript or not. And if we make a transcript, how much transcript should we make? One video today, so I want you to sequence the lac and the trip operons and how they are different from each other, and talk a little bit about what turns things on and off in bacteria, why do they care, and what's the difference between repressible and inducible. So some background, and I pulled this out of two different parts of the textbook. I'm only holding you to chapter 16 in this case, but I pulled a couple things out of somewhere else, and I'll show you where I did that. But when bacteria make their genes, some of them are genes that they need to use all the time. They need the products of those proteins to function on everyday life. Those things are called constitutive genes or constitutive enzymes. They are always on and always produced. Other parts of the genome for bacteria can be turned on or off depending on whether it's appropriate to make them. The goal here is for the bacteria to never waste energy. Energy wasted is energy that was not used to make baby bacteria, and pass those genes on. So bacteria have evolved to be incredibly efficient with the products and the products they make and the energy that they produce. They do that by regulating their gene and their protein production with a series of switches. Eukaryotics are different. We're going to do that in a different mechanism. We're going to have genes that are inducible in bacteria. They can be turned on, but their normal state is being off. If I have a molecule that arrives in the bacteria's experience, either outside or inside the bacteria, it will trigger transcription to start. I can induce it. I can make it start. I often, if I want to make enzymes that are coded for in an inducible piece of code, I need an inducer. This is a noun. That's not how you spell noun. A repressible section of the code can be turned off, but it starts out as on. The normal state of it is to make that product. The presence of a molecule turns it off. So if the repressor, again, a noun, this is a molecule, is in the environment of the bacteria, it stops making transcripts of this gene. Often the repressor is the thing that the gene itself codes for. So it has no beta-galactosidase. That's a terrible example. It has no molecule that it needs to make. And then as that level of that molecule increases, the presence of that molecule says we don't need it anymore. We've made enough, and it shuts down the system. I can repress it. We also have two things that are different concepts, but ways that we control this from outside. Bacterial and just transcription in general can be controlled positively. It only works when a regulator molecule stimulates RNA production. So I need a molecule to keep it going. This is kind of like the accelerator in your car. You have to keep pushing the accelerator to make your car go further and faster, or even to go. If you don't have your foot on the accelerator, unless there's something wrong with your car, it doesn't go anywhere. It shouldn't just go without you pressing anything. Negative controls are where the transcription occurs unless something is shut off. It's like the brake in your car. If you have your foot on the brake, it doesn't matter how much you push on the accelerator as long as your car is working. Your car doesn't go forward. Having this regulator molecule, your foot on the brake, stops transcription. So I put this little chart here from a different place in the text, or different text altogether, actually. I think this is a handy-dandy guide. If the protein is present, and we're using positive control, the operon, the unit of transcription, is on. If the regulatory molecule is present and we're using negative control, that operon is off. There's a homework that you might want to write this out to solve. This will help you solve the homework where you have to drag and drop the different kinds of molecules. Inducible means the molecule's presence starts the transcription. Repressible means the molecule's presence stops transcription. Positive control is a factor or some kind of molecule, not the same one, that activates. Negative control means that we can have the presence of a molecule stops it from happening. So these two go together, and these two go together. The operon is a group of genes in bacteria that are controlled by a single on-off switch. It's called a regulatory signal. That is called the operator. This is something that is only found in prokaryotes. You do not have operons. Oops, I thought the picture would appear here. The operon is a series of genes all tied together, an operator that has a switch on it, a segment of DNA with a switch on it, we call the operator, that allows you to transcribe those genes or prevents you from transcribing it, and a promoter region where RNA polymerase can attach. The genes on the operon are transcribed together. They are polycistronic. We've introduced that word a couple different ways. This is where it really matters. If you make one of the genes in an operon, you make all of the genes in the operon. They are then later cut apart either during translation or they just produce different proteins. They're all individually translated by different ribosomes at the same time. An operon is a sequence of DNA we're transcribing. I have a promoter. This is the P region here. I have an operator where there is going to be a switch to turn on and off the gene. It's going to do that by either being blocked so that nothing can, RNA polymerase can't move from the promoter to the genes, or by not being blocked. If the operator is free, the RNA polymerase can pass through the operator with ease and then transcribe these genes. This operator is named after the operator from like the 1940s, the woman who would sit at the phone company and people would call her and she'd actually plug plugs into holes and connect you directly with someone. The operator is either allowing you to pass through as RNA polymerase or not allowing you to pass through. The promoter and the operator are binding sites. They are generally not transcribed. So this is not part of a protein. This is not going, they might be, parts of them might be translated, but they never get, they never get transcribed. They're not translated. So this is from your textbook. I would like you to know beta-galactosidase because we're going to talk about it. We talked about it in chapter 20 when we did that last week. But I'm going to give a general idea of how they work. I am also really interested in you knowing what all these different color parts mean. In prokaryotes, all genes are regulated at transcription. The lac operon is inducible. That means it is normally off. You can turn it on if lactose is present around the cell. These three genes, beta-galactosidase, permease, and transacetylase, are all involved in lactose metabolism. This is not how you work. This is not, if you are lactose intolerant, this doesn't have anything to do with you. That's a different story. But this is how E. coli bacteria deals with the presence of lactose in its environment. If there is no lactose, the cell does not make any of these enzymes. Holy cow, that would be a waste of energy. All of these are just involved in breaking down lactose. If there is lactose, the presence of lactose acts as an inducer. It allows the RNA polymerase to pass through the operator and actually transcribe these three genes. The three genes, I have them on two different slides, or three different slides, are permease here. Permease facilitates lactose coming into the cell. So if lactose is exposed to, the E. coli is exposed to lactose, the cell starts looking for lactose outside. It creates a transmembrane protein, permease. Transacetylase, we think this actually cleans up the lactose metabolism byproducts, but I don't have a solid answer for you on that yet. So we're going to just leave this as It's definitely important in lactose metabolism. Beta-galactosidase, which we talked about in chapter 20, beta-galactosidase was involved in the blue-white test. If we were trying to determine whether we inserted use the restriction enzymes to insert the gene correctly or not in the plasmid, beta-galactosidase is an enzyme who breaks lactose into glucose, which is a monomer, and galactose. Lactose is a dimer. A disaccharide, sorry. It's a disaccharide. There are 2 monomers involved in lactose. When lactose is exposed to beta-galactosidase, it breaks those 2 monomers in half. This glucose is what the cell really, really loves. E. coli prefers glucose to lactose. No lactose, only glucose. It's not energy efficient to break down lactose. You have to use energy as a cell to break the lactose in half to get a little bit of glucose out of it. Glucose is preferable. So we are going to have this situation where even if I have presence of lactose in my surrounding environment as an E. coli cell, I still want to prioritize glucose. I'm going to use negative control to transcribe when the repressor is missing. So if lactose is present, I want to use my lactose metabolism a little bit. Normally, since there is no lactose normally in the E. coli environment, and because glucose is better, normally the repressor is blocking the operator, and all 3 of those genes are not transcribed. Now, one more little guy here. This upstream of the lac operon, and not exactly part of the lac operon, but it plays a role in it, is LAC-L. This LAC-L produces a repressor, and it's an allosteric repressor, meaning it can change shape. It's from a different location. It causes a change in the operator itself. This repressor protein normally binds to the operator and blocks it. We know there are actually multiple repressor sites. We think that site 1 and site 3 need to be blocked in order for this process to be completely blocked. There is a site 2. As far as I could tell, we don't have a solid answer on what site 2 actually does yet. But the overall function of this is the LAC-L makes this repressor. This repressor binds to the operator. I'll make it like right here. And then when RNA polymerase attaches to the promoter and attempts to transcribe, there's literally a physical object in its way, and it can't transcribe. If lactose is present, lactose tells the cell that it should maybe need these enzymes. So what happens is lactose becomes the thing that triggers turning on the transcription of this gene. Lactose is the inducer. Lactose binds to the repressor, and when it binds to the repressor down here, when lactose binds to the repressor cannot block the operator. It's the wrong shape. And so the promoter and all of the RNA polymerase that binds to the promoter is free to transcribe. If there is no lactose, the repressor gets made and the repressor binds to the operator and we can't transcribe. And this is normal. This is if lactose. If the lactose gets all used up and we break it into glucose and galactose, there is no more lactose and we will stop blocking the repressor molecules. They are consistently made. They are constitutively made. When we run out of lactose, this operon gets blocked again. So it's kind of genius if you think about it, because this cell isn't smart enough to be like, hey, lactose, I should change my role. The presence of lactose triggers the ability to transcribe these genes. The absence of lactose prevents the transcription of these genes. If lactose is present, Now I got to decide how much do I want to actually transcribe, because just transcribing like crazy means I'm wasting energy. I want to transcribe exactly enough transcript to deal with the lactose that I have without wasting any energy. So there's two more molecules that we need to talk about here that control the positive control, or the sort of how much do I actually need to make of this situation. There's a molecule inside of your cells and inside of bacterial cells called cyclical AMP, or cyclical adenosine monophosphate. This is a byproduct of breaking ATP down and taking two phosphates off of it, and you leave an AMP, an adenosine monophosphate. This version of it is cyclical. It is manufactured by a protein or an enzyme inside the cell called adenyl cyclase. Glucose inhibits adenyl cyclase. So this thing that makes cyclical AMP can't make cyclical AMP if glucose is there. If there is a lot of glucose, there's very little cyclical AMP. If there's little glucose, or no glucose, there's a lot of cyclical AMP. Catabolite activator protein, I'm going to go on to the next slide, or CAP, is activated when cyclical AMP binds to it. Cyclical AMP rises as glucose levels go down. For every cyclical AMP that is freed, each one binds to a molecule called catabolite activating protein, or the protein that activates the breaking down of some other substances. Catabolite activating protein, CAP, binds to the CAP binding site at the back of the promoter region. It's literally binding to the DNA. And as RNA polymerase binds to the area of the DNA that is going to allow it to pass through the operator and transcribe, CAP stabilizes RNA polymerase. If CAP isn't here and RNA polymerase binds and transcribes, in the case of very high glucose, RNA polymerase is pretty wobbly and it will actually regularly fall off this site. This is a feature, not a bug. RNA polymerase is not very good at its job unless somebody holds on to it like a toddler learning how to ride a bike. CAP, when it's bound to the CAP binding site, stabilizes RNA polymerase and makes it more efficient at transcription. The lower the glucose level is, the higher cyclical AMP, the higher CAP is active. I'm going to write active CAP. And this leads to high efficiency of transcription. And the opposite is also true. If there's high levels of glucose, there's low levels of cyclical AMP. Very few, if any, CAP molecules get activated, and so the transcription remains very, very low, which is great because we prefer glucose as E. coli over lactose. To sum this up in a very different way, in a simpler version, if there is no lactose, the lac operon is off. The repressor is bound to the operator. That repressor is made constitutively, so it's always being made. The promoter, if RNA polymerase binds to it, cannot pass through the operator. There is no transcription. He's got lots of glucose, so he's happy. If lactose is present, lactose acts as an inducer. It induces by removing the repressor from the operator. So for every lactose molecule, it deactivates one repressor. RNA polymerase can bind to the promoter, and it makes its way through the free operator and can transcribe. If lactose is present, we also now have to ask the question, how much glucose? Because glucose is going to determine how efficient I want to be at transcribing my lac operon as an E. coli. If there is a lot of glucose, I don't want to be good at this. I use the lactose, but I don't want to use energy if I have an abundant supply of glucose to break down lactose. That's wasteful. So if there's a lot of glucose, my cyclical AMP and my active cap will therefore be low, and my transcription rate will be low. If there's low or no glucose, my cyclical AMP is being made by adenyl cyclase because glucose is not there to inhibit it. My cap is active because I have a lot of cyclical AMP binding to that catabolite activator protein, and so my transcription rate is much higher. In this case, because as soon as you have lactose, you now have to ask the question, how much should I make? I have a couple practice questions with this, and we can work in this in lab too. This operon model, the way to get around where it might be starting to get confusion is, remember, we want to be as energy efficient as possible. Evolution has driven energy efficiency in this case. So every outcome is going to waste as little energy as possible, unless there's something wrong with your bacteria. I want to compare the lac operon to the trip operon. This is an operon that makes tryptophan. It is repressible if there is tryptophan in the environment for this bacteria. It does not need to make it. If tryptophan exists, it represses the trip operon. If there is no tryptophan in the environment for the bacteria now needs to make it. When it makes it, it only needs to make it if it's not already there. So it will, if it is not repressed, its normal function is to just keep making this tryptophan molecule. So if tryptophan is absent, E. coli needs to make it. If tryptophan is present, E. coli doesn't need to make it. So kind of makes sense, right? If you have dinner and it's already prepared, you don't need to make dinner. If you come home and there's no dinner, you need to make it. It's literally the same choice, except it's being controlled by a series of molecules that are grabbing onto the DNA region that is coding for these proteins and either allowing transcription or not allowing transcription. In the absence of tryptophan, an inactive repressor is made, but it can't actually bind to the operator. So we make this repressor. Now I'm getting into the details of it. The repressor is made, but it can't bind to the operator. When tryptophan is present, it binds to the repressor, and it causes the change that makes the repressor rate shaped to the operator, and it actually blocks it. The repressor gene is constitutive. It is always being made, but it's making a gene that is off version. If tryptophan is there, we want to block the operator. So I take this version, the off version plus actual tryptophan, blocks the operator. I don't need to make it because tryptophan is already existing in my environment. I'm a bacteria. There is a leader sequence. It's called I would call it an attenuator. Your book calls it both, but I would use an attenuator. Attenuation, the leader sequence is actually transcribed, even if the operon is repressed. What it actually does is it's a sequence that folds into hairpins. We have seen this before, where it actually has a series of matching nucleotides that will tend to form hydrogen bonds with each other and create a hairpin curve, or something that interferes with the molecular function of the other molecules. Here, better than my drawing. This terminator hairpin, or an anti-terminator hairpin, it's actually caused by a series of codes of nucleotides, and they will form hydrogen bonds with each other, forming these really characteristic turns and shapes. This actually will cause ribosomes to stall if they're trying to actually translate this, so that they're unable to do it. The anti-terminator hairpin is made if there is no tryptophan. The terminator hairpin is made if tryptophan is present. If the terminator hairpin is made, we're going to translate. If the anti-terminator hairpin is made, we are not going to translate. I don't know if these happy faces are how tryptophan feels about this, I guess. So a little bit of questions. Just, I know that this is a short lecture. It's a lot in a little lecture. I can go over this with you again in lab. I think that the trick to this is to treat it like a computer program. It is either on or off, or it has a series of switches. They are not smart. They are making decisions based on the existence or absence of a molecule. The lac operon is optimally expressed blanky blank blank blank. Optimally means expressed as much as possible. Let me go on. Lactose, no glucose is going to, the presence of lactose is going to remove the block from the operator. We're going to induce the operon. And the lack of glucose means that there's really high levels of cyclical AMP that activates a lot of CAP, that stabilizes RNA polymerase and allows transcription to occur as efficiently as possible. And my rationale over here for you. And my other question, a little bit to think about, this is an imaginary operon. P is the promoter. A is the first gene. Z is the last gene. 1, 2, 3, and 4 are places where I can insert something. Where is the operator if I was going to try to, I'm going to turn either 1, 2, 3, or 4, I'm going to designate as the operator. Should the operator go before the promoter, after the promoter, or in between, or after the genes? I want the promoter region to be where RNA polymerase binds. RNA polymerase has to pass through the operator. If the operator is free, RNA polymerase can proceed and transcribe. If the operator is blocked, RNA polymerase cannot pass through and cannot transcribe. Kind of as a side note, where does the CAP binding site go? Does it go before the promoter, after the promoter, or in the genes? Here, CAP binding site is going to be just at the beginning of the promoter. Very nice. All right, well, with that, I'm going to wrap that up. I'll help you as we go through with the operons. I feel like many of you said that you had not covered this before, so we can cover it again in a little bit. It's just a logic game. Don't overcomplicate it, like truly. Compared to some of the other stuff we talked about, this is very, very logical because it's just evolutionary driven efficiency. And I'll see you next week in lab.